Assalamu alaikum class. Welcome to our first online session, and you all know that I'm your biochemistry teacher, Dr. Bushra Ijaz, and I'll be teaching you principles of biochemistry. The course code is Bio 132, and you all know that we have previously discussed about the credit hours and the content, but let's just briefly do that again. So here is the main course content that we will be discussing online. We're going to divide it into parts, and we are going to decide about the content that we will add into your session number 1 and session number 2 and so on. So in today's lecture, we will be looking at the foundations, the biochemical foundations of life. We will look at the cellular, chemical and physical foundations along with some genetic foundations, biochemical foundations of life. Then we are going to discuss about the biochemistry of water. We are going to look into detail the chemistry of water, the bonds that it is able to make, and how the presence of the lone pair or the bonding pair. Well, I'm going to discuss what lone pair and bond pair are. We are actually going to discuss some basic chemistry so that you can understand how these biological molecules, for example, water or the most important carbon molecule, is able to behave in a certain way. After doing that, we are going to start with the protein chemistry. We will look at the structure of protein, the basic elements, amino acids, peptides, and proteins, their three-dimensional structure, as well as we're also going to look at some of the classes of protein tertiary structure, which are the globular and fibrous proteins, along with the enzymes. And we are going to discuss their biochemical role and some of the main functions that they are actually performing inside the cells. Then, an important topic is the study of intermediary metabolism. We will be looking at the bioenergetics and oxidative phosphorylation along with some basic concept that you need to know in the cycle that you need to learn. We're going to look at the carbohydrates, which are actually it's going to be a very important topic that we will be discussing. And to understand carbohydrates, we need to understand the biochemical nature and the chemistry of carbon and hydrogen molecule, along with the other nitrogen and phosphorus molecules that are actually important for all the important tasks carbohydrates are able to perform. We're going to look at glycolysis, TCA cycle, gluconeogenesis, and glycogen metabolism, along with the metabolism of mono and disaccharides. We will also look at the pentose phosphate pathway and also the production of NADPH. We are going to study the chemistry of ATP molecules, NADP molecules, FAD molecules, and NADPH molecule in particular. We will look at the aminoglycans, proteoglycans, and glycoproteins. We will also look at the biochemistry of lipid and their metabolism and metabolism of dietary lipids, fatty acids, triglycerols, cholesterol, and steroids. So let's formally start our course of Principles of Biochemistry with our focus on the foundations of biochemistry. Now, to understand the biochemical foundations of life, we will start from the scratch and we will learn how life actually began. We'll first look at the first elements and biomolecules, and then we're going to move on to some complex structures. Now, to understand the foundations of biochemistry, I found a very interesting video to start with. So let's just watch it.
Stars, like our own sun, form from gas clouds that have about every kind of element there is, as well as some pretty complicated molecules. Stuff left over after the sun forms usually turns into a system of planets. Some of these planets have a solid surface, perhaps liquid seas, and a gaseous atmosphere. A rich environment for atoms and molecules to come together in a complex chemical reaction. Carbon atoms in particular link together to form complex organic molecules and amino acids. Chemical catalysts speed reactions along. The products of one chemical reaction become the raw material for new reactions. At some level of complexity, a catalyst becomes an enzyme. An amino acid chain becomes a protein. A loop of chemical reactions become a metabolism. And chemistry becomes biology. On our planet, and perhaps countless others, life arose. The DNA molecule, which is the basis of all organic life on Earth, is more intricate by far than any spiral galaxy. Because the structure of DNA contains something new, something that was missing from inanimate matter before the origin of life. It contains information. The DNA molecule encodes not only the information necessary to make copies of itself, but the information necessary to construct an entire organism. The blueprints for an ant, or a dolphin, or a bullfrog, or even a person. All of this information is somehow built into the structure of an organism's DNA in a molecular code billions of letters long. This information wasn't always there. The first DNA molecules could probably do nothing more than make copies of themselves from the surrounding molecules. This self-reproduction is a fundamental aspect of what we call life. And once there was life, a new set of rules took over, the laws of natural selection. Life became matter that evolved as a result of environmental pressures, matter that changed randomly. But those changes only endured if they improved a life form's chances of survival in each environment. Change with a purpose. While a species' evolution is determined by the interaction of random changes and environmental pressures, changes in an individual life form can be the result of an organism's reacting and adapting within its own sphere of influence. It can push back at its environment. The better a system was at making copies of itself, the more copies there were in the next generation. As the first living organisms adapted to ever-changing environments, a greater diversity of living things arose. A side effect of this increase of diversity was increased complexity. Now, all of our tests point ever more towards the complex end of our complexity meter. Consider a living single-celled organism. There's nothing reversible about life or death, or eating or being eaten. And there is uniqueness. Unlike electrons that are interchangeable, even single cells are absolutely unique. They may be capable of the same functionality, but they are always different in form and structure. Single cells soon join together to form multicellular colonies. And cells in these new colonies, once functionally interchangeable, became increasingly specialized. Some cells did the digesting, others became responsible for movement. Specialized cells led to the specialized tissues and organs. And most importantly, cells that could respond to light, or electricity, or chemical disturbances evolved into full-fledged nervous systems.
That was quite understanding, and that was quite interesting to see that how life actually began and how these basic elements were formed. So, the main objective of today's lecture and the main objective of our course is actually understanding biochemistry, which focuses on understanding the chemical bases which allow biological molecules to give rise to the processes that occur within living cells and between the cells. And these processes then in turn relate greatly to the study and understanding of tissues and organs as well as organisms, structure and function. As you have already seen in the video that I have embedded in this video lecture, in which we learned about the Big Bang Theory and the formation of the first elements and life on Earth and so on. Let me just briefly explain that to you again in very simple words. It's just a story that you need to learn. You don't need to like memorize it for the sake of exam. It's just to make it interesting. You all know that 15 to 20 billion years ago, the universe came across an eruption of hot, energetic, rich subatomic particles. And then within seconds, the simplest elements, which were hydrogen and helium, they were produced. Now, as the universe expanded and cooled, materials condensed under the influence of gravity to form stars. Some stars became enormous and then they exploded and resulted in the formation of supernovas, and they released a large amount of energy, and that led to different type of reactions, fusion reaction or fission reaction. Now these reactions are actually, this energy is actually required for the fusion of simple atomic nuclei into more complex element. Thus, were produced over billions of years ago, the earth itself and the chemical elements found on the earth today. About 4 billion years ago, life arose as simple microorganisms with the ability to extract energy from the organic compounds or from the sunlight, which they use to make a vast array of more complex molecules from the simple element and compounds on the Earth's surface. Now, biochemistry actually asks how these remarkable properties of living organisms arise from thousands of different lifeless molecules like carbon, hydrogen, nitrogen. When these molecules are isolated and they are individually examined, we are able to know that all of them come from physical and chemical laws that describe the behavior of non living inanimate matter. So, as do all the processes occurring in the living organism, they are able to perform them. Now, the study of biochemistry actually shows how the collection of these non living molecules or inanimate molecules that constitute the living organism, how they interact to maintain and to prosper the life, and which is like actually by the physical and chemical laws that govern the non living universe. Yet the organism possess extraordinary attributes and properties that distinguish them from other collection of matter. Now, what are these distinguishing features of the living organism which separate them from the inanimate universe? Let us just briefly look at some of the main features of the living organism which separate them from the inanimate universe. You all know that all the living organisms they have a high degree of chemical complexity and they have microscopic organization. Thousands of different molecules make up a cell's intricate internal structure. Each has its characteristic sequence of subunits, its unique three-dimensional structure, and its highly specific selection of binding partners in the cell. Secondly, you all know that they all have a system for extracting, transforming, and using energy from the environment, which actually enable them or the organism to build and maintain their intricate structure and do mechanical, chemical, osmotic, and electrical work. Inanimate or non-living matter tends rather to decay towards a more disordered state to come to equilibrium with its surrounding. Another important feature of the living organisms is that they have this capacity for replication. They are able to replicate or they have the ability to have themselves self-assembled in a precise way. For example, a bacterial cell, when it is placed in nutrient media, which can give rise to a billion of its copies, which are actually identical to itself within 24 hours. Each cell contains thousands of different of molecule, and some are extremely complex. Yet, a bacterium is faithfully able to copy itself and form an original copy from the original one, and its construction directly entirely form information contained within the genetic material of the original cell. Living organisms also have this mechanism for sensing and responding to the alteration in their surrounding environment, and they are therefore able to constantly adjust to the changes, or they are able to adapt to their internal chemistry. Living organisms also have a defined function for each of their component and regulated interaction among them. This is true not only for the macroscopic structures such as leaves and stems or hearts or lungs, but also for the microscopic intracellular structures and individual chemical compounds. The interplay among the chemical components of the living organism is dynamic, and changes in one component causes coordination or compensation changes in another, with the whole assembly displaying a characteristic which is beyond that of its individual part. Along with that, you all know that all the living organisms have a history of evolution. I mean, they have been evolving over time. Organisms change their inherited life strategies to survive in new circumstances. That's actually the idea that was represented by Darwin, which is survival of the best. The result of these changes or evolution in an organism leads to an enormous diversity in life forms, which are very different but fundamentally they are all related through some shared ancestry. Along with all those features, we all know that diverse living organisms have common chemical features when we look at them biochemically. For example, if say it is a bird or a beast, a plant, or a soil microorganism, they all share something with the humans in their basic structural units or the cells and the kind of macromolecules that they have. For example, their DNA, RNA, and proteins. They're all made up of same kind of monomeric units, which are actually nucleotides and amino acids in case of DNA and protein. They are able to utilize the same pathways for synthesis of cellular component, and they share the same genetic code and derive from the same evolutionary ancestors. Biochemistry actually describes in molecular terms the structure, mechanism, and chemical processes shared by all the living organisms and provide organizing principles that underline life in all of its diverse form. Principles were actually referred to collectively as a molecular logic of life. Although biochemistry provides important insights and practical application in medicine, agriculture, nutrition, and so on, but its ultimate concern is with the wonder of life itself. In this introductory lecture, I will be describing actually briefly the cellular, chemical, physical, and also the thermodynamic as well as genetic background to for all the principles that are involved in evolution and the development of generations of properties of living cells. And as we move on with this course, you will find it helpful to refer back to this lecture at interval to refresh your memory of the background material. Now, to understand the molecular logic of life, let's start with the cellular foundation. The unity and diversity of organisms become apparent even at the cellular level. The smallest organisms consist of a single cell and they are microscopic in nature. Large multicellular organisms contain many different types of cells which vary in size, shape, and the function that they perform. Now, despite these obvious differences, all cells of the multicellular organism contain many different types of cells, and they are all simple and the most complex organisms share certain fundamental properties which can be seen at the biochemical level or the chemical level. Cells of all kinds share a certain structural feature. For example, you all know they are all covered with plasma membrane, which actually defines the periphery of the cells and separates its content from the surrounding. It is composed of lipids and protein molecules. You have all learned about the structure of the plasma membrane. You know it's thin, it's tough, it's elastic, it is a kind of hydrophobic barrier around the cells. The membrane, which is, if I define it as a barrier for the free passage of inorganic and most of the charged or polar compounds, transport of proteins in the plasma membrane allows the passage of certain ions and molecules. Therefore, it is acting actually as a sieve, a permeable, semi-permeable sieve, and it also has receptors which are able to like transmit or receive the signals into the cells. You know that these are also there are certain enzymes which are present in the plasma membrane, and there are certain reaction pathways that are taking place there. Because the individual lipid and proteins of the plasma membranes are not covalently linked, they are just embedded in the membrane, the entire structure is remarkably flexible and that allows changes in the shape and size. That's why we are growing. The internal volume bounded by the plasma membrane is called the cytoplasm, you know that, and it's composed of aqueous solution. It has mostly water and then it's called cytosol. And it has a variety of suspended particles and they are all performing different functions. Cytosol is, you can say that it's highly concentrated solution. It contains enzymes and the RNA molecule that encode them. The components such as amino acids and nucleotides, which of these are like macromolecules and they are assembled, they are going to form hundreds of small organic molecules, which are called metabolites. And there are some intermediates in the biosynthesis and oxidative degradation pathways. And it also has coenzymes, which are the site of the protein synthesis. Now, along with the plasma membrane and cytoplasm, all the cells have, at least for some part of their life, they either have a nucleus or a nucleoid, which contains the genome. And you all know that genome is the complete set of the gene, which is composed of DNA, which is stored and as well as it's able to replicate. The nuclide in the bacteria is not separated from the cytoplasm by a membrane, you know that, and the nucleus in higher organisms consists of nuclear material enclosed within double membrane, which is also called the nuclear envelope. Cells with a nuclear envelope, they are called eukaryotes, and the cells which don't have this nuclear envelope, for example, the bacterial cells, for example, E. coli, they are all called prokaryotes. Most cells are microscopic, which means that they are invisible to us with unaided eyes. And animal and plant cells are typically they are larger in size, and their size in terms of diameter is around 5 to 100 meter in diameter. And many bacteria, which are only 1 to 2 nanometer long. Now, you need to understand that what thing actually decides the size or dimension of the cell. There are two limits. There is this lower limit, which is probably set by the minimum number of each type of biomolecule that are actually required by the cell. The smaller cell, for example, certain bacteria, they can be like 300 nanometer in diameter and they have a volume of about 1014 ml. They're really small in size. A single bacterial ribosome is about 20 nanometer and it's the its longest dimension. So for a few ribosomes to take up a substantial fraction of the volume in a bacterial cell. Now, the upper limit of the cell size is probably set by the rate of diffusion of the solute molecules in aqueous systems. For example, a bacterial cell that depends upon oxygen consuming reactions to for the production of the energy, it must obtain molecular oxygen by the process of diffusion from the surrounding media through its plasma membrane. The cell is so small and the ratio of its surface area to its volume is so large that every part of its cytoplasm is actually easily reached by the oxygen which is diffusing into the cell. As the cell size increases, however, surface to volume ratio is going to decrease until the metabolism consumes oxygen faster than the diffused supply that it has. Metabolism that requires oxygen thus becomes impossible as the cell size increases beyond a certain point, placing a theoretical upper limit to the size of the cell. Let's just briefly look at different domains of life. You all know that all the living organisms fall into three large groups, or we can also call them kingdoms or domains, that actually define three branches of evolution from a common ancestor. The two large groups of the prokaryotes can be distinguished on biochemical background. For example, we have archaebacteria and we have eubacteria. Eubacteria inhabit soil, surface of water, and the tissues of other living organisms or decaying organisms. You have all those eubacteria all over your body. Most of the well-studied bacteria, including E. coli, are eubacteria. The archaebacteria, more recently, they are discovered as compared to the eubacteria, and they are less well characterized biochemically. Most of them are present in extreme environments, for example, in salt lakes or hot springs or in highly acidic bogs and ocean depths. The available evidence suggests that the archaebacteria and eubacteria actually diverged early in evolution and constituted two separate domains. Sometimes they are also called archaea and bacteria. All eukaryotic organisms, which make up the third domain, which is also called the eukarya, evolved from the same branch, but they gave rise to the archaea or archaebacteria, and therefore more closely related to eukaryotes than to eubacteria. Within the domain of archaean bacteria are some subgroups which are distinguished by the habitat in which they live. For example, in aerobic habitat with a large supply of oxygen, some resident organisms derive energy from the transfer of electrons from the fuel molecule to the oxygen. Now, how this happens, how the transfer of electron is able to produce energy, we are going to study that in detail. Other environments are anaerobic environment in which there is less supply of oxygen, limited or no supply of oxygen, and the microorganisms are therefore adapted to these environments, and they obtain energy by transferring electron to the nitrate or the sulfate or the carbon dioxide molecules, and they would form nitrogen, hydrogen disulfide, and methane gas, especially. Many organisms that have evolved in anaerobic environment they are obligate anaerobes, which means that they are going to die when they are exposed to oxygen. We can classify organisms according to how they obtain energy and carbon they need for synthesizing cellular material. And there are two broad categories based on energy sources. They are either called the phototrophs, which means that they are able to take nourishment from the light by using the sunlight, and then there are chemotrophs, which are able to derive or they are able to obtain their energy from the oxidation of fuel. What is oxidation and reduction reaction? I hope you know that we are going to discuss them in detail as we move on to our course. Now, all the chemotrophs require a source of organic nutrients. They cannot fix the carbon dioxide into organic compounds. The phototrophs, on the other hand, they can be further divided into the ones that can obtain all needed carbon from carbon dioxide, which are therefore called the autotrophs, and those that require organic nutrients, which are called therefore the heterotrophs. No chemotroph can get its carbon atom exclusively from the carbon dioxide. That is, no chemotroph are autotroph, but the chemotrophs may be further classified according to different criteria, whether the fuels that they are oxidizing are organic. If it is organic, they are called organotroph, and if it is inorganic, they are called the lithotrophs. Most known organisms fall within one of these four broad categories: autotrophs or heterotrophs among the photosynthesizers, lithotrophs or the organotrophs among the chemical oxidizers. The prokaryotes have several general modes of obtaining carbon and energy. For example, we have E. coli. It is an example of chemo-organo-heterotroph, which means that it requires organic compound from its environment as a fuel and as a source of carbon. Cyanobacteria are photolithoautotroph, which means that they are, because the word photo is there, they are able to trap the sunlight and they are able to use it as an energy source and convert carbon dioxide into biomolecules. So, what are we humans? We humans, like E. coli, are chemo-organo-heterotrophs. Now let's look at E. coli, which is the most studied prokaryotic cell. You all know that bacterial cells share common structural features, but they also show group specification or specialization. E. coli, for example, is usually harmless and it is in the human intestinal tract. E. coli cell is about 2 micrometer long and it has less than 1 micrometer in diameter. It has a protective outer membrane and an inner plasma membrane that encloses the cytoplasm and the nuclei. Between the inner and the outer membrane, there is a thin but a very strong layer of a polymer which is called peptidoglycan, and this peptidoglycan actually gives the cell its shape and rigidity. The plasma membrane and the layers outside of it constitute the cell envelope. You see that, and in the, for example, in the archaea, the rigidity of the envelope is conferred by a different type of polymer, which are called pseudopeptidoglycans. The plasma membrane of eubacteria consists of a thin layer of lipid molecules which is actually penetrated by the protein. Archaebacterial membranes have a similar structure, and although their lipid content, which is strikingly different from those of the eubacteria. The cytoplasm of E. coli contains about 1,500 ribosomes, thousands of copies of each of about 1,000 different enzymes, numerous metabolites and cofactors, and a large variety of inorganic ions. The nucleide contains a single circular DNA, and the cytoplasm, like the cytoplasm of most of the bacteria, it contains one or more smaller circular segments of DNA called the plasmids. In nature, some plasmids are responsible for giving resistance to the toxins and antibiotic in the environment. In the laboratory, these DNA segments are specially used for the sake of experimental manipulations, and they are very useful to study different molecular genetics. Most bacteria, for example, also E. coli, they have lead to existence such as individual cells, but in some bacterial species, they tend to live in clusters and filaments, and a few of them, for example, a myzobacteria, for example, they demonstrate simple social behavior. On the other hand, when we look at the eukaryotic cells, they have a variety of membranous organelles which can be isolated for the sake of study. Look at the figure and you see that there are typical eukaryotic cells, and they are much larger as compared to the prokaryotic cells. They are 5 to 100 micrometer in diameter with a cell volume which is thousands to million times larger than that of a simple bacteria. The distinguishing characters of each eukaryote are the nucleus and a variety of membrane-bound organelles with specific functions. For example, mitochondria, endoplasmic reticulum, Golgi body, and lysosome. You all know and you have already studied about them. Plant cells also contain vacuoles and chloroplast, as you can see in this figure, and they are present in the cytoplasm. Many cells are granules or droplets that contain stored nutrients such as starch and fat. Also present in the cytoplasm of many cells are major biochemical molecules which are responsible for containing the biochemical life or the energy producing activities. Now, an essential step towards the identification and isolation of these biomolecule was the discovery of process of fractionation. In a typical process of fractionation, cells or tissues in solutions, they are actually disrupted by homogenization, which is quite gentle, and this treatment ruptures the plasma membrane but leaves most of the organelles intact. The homogenate is then centrifuged, and the organelles such as nuclei, mitochondria, and lysosomes, because they are different in size and therefore they have different sedimentation constant, they're going to sediment at different rates. They also differ in their specific gravity, and they are therefore going to float at different levels in a density gradient. Differential centrifugation results in a rough fractionation of the cytoplasmic content, which may be further purified by the same density centrifugation. In this procedure, organelles of different buoyancy, the organelles, they are then separated and then they're further studied. For example, these methods were actually used to establish that lysosomes contain degradative enzymes, mitochondria contain oxidative enzymes, and chloroplasts contain photosynthetic pigment. This isolation of the organelle, actually enriched in a certain enzyme, is often the first step in the purification of that enzyme. Now, when we talk about the macromolecules and their monomeric subunits, they differ greatly in size. You all know that there is a structural hierarchy. A molecule of alanine, for example, alanine is an amino acid, we're going to study that in detail. It is less than zero point 0.5 nanometer long. Hemoglobin molecule, which is the oxygen carrying protein of the eukaryotes or the red blood cells, it consists of nearly 600 amino acid subunits in four long chains, which are folded into globular shape, and they are associated in structure, which is around 5.5 nanometer in diameter. In turn, proteins are much smaller than the ribosomes, which are about 20 nanometer in diameter, and in turn, much smaller than the organelles, such as mitochondria, typically, which is around 1000 nanometer in diameter. So it is a long jump from a simple biomolecule to cellular structure that can be seen within the light microscope. The monomeric subunits in proteins, nucleic acids, and polysaccharides are joined together with the help of covalent bond. When we will study about the carbon and its chemistry and its stereochemistry, I'm going to describe and give you different examples of different type of bond which are present within the biomolecules. And we're going to study briefly what are different types of bond and which of them are really important for us. Now, in supramolecular complexes, however, the macromolecules are held together by non-covalent interactions, which are much weaker individually as compared to the covalent bond. Among these non-covalent interactions are hydrogen bonds between polar groups, ionic interactions, which are between charged groups, hydrophobic interactions, which are among the non-polar groups in aqueous solution, and van der Waals interactions. And all of these interactions have energies substantially smaller than those of the covalent bond, which is the true or the strongest bond. The nature of these non-covalent interaction will be described later in the chapter where we will be studying about the chemistry of carbon. The large number of weak interactions between macromolecules in supramolecular complexes stabilize these assemblies, producing their unique structures. Now, when we talk about the macromolecules and their monomeric subunits, they differ greatly in size. You all know that there is a structural hierarchy. A molecule of alanine, for example, alanine is an amino acid, we're going to study that in detail. It is less than zero point 0.5 nanometer long. Hemoglobin molecule, which is the oxygen carrying protein of the eukaryotes or the red blood cells, it consists of nearly 600 amino acid subunits in four long chains, which are folded into globular shape, and they are associated in structure, which is around 5.5 nanometer in diameter. In turn, proteins are much smaller than the ribosomes, which are about 20 nanometer in diameter, and in turn, much smaller than the organelles, such as mitochondria, typically, which is around 1000 nanometer in diameter. So it is a long jump from a simple biomolecule to cellular structure that can be seen within the light microscope. The monomeric subunits in proteins, nucleic acids, and polysaccharides are joined together with the help of covalent bond. When we will study about the carbon and its chemistry and its stereochemistry, I'm going to describe and give you different examples of different type of bond which are present within the biomolecules. And we're going to study briefly what are different types of bond and which of them are really important for us. Now, in supramolecular complexes, however, the macromolecules are held together by non-covalent interactions, which are much weaker individually as compared to the covalent bond. Among these non-covalent interactions are hydrogen bonds between polar groups, ionic interactions, which are between charged groups, hydrophobic interactions, which are among the non-polar groups in aqueous solution, and van der Waals interactions. And all of these interactions have energies substantially smaller than those of the covalent bond, which is the true or the strongest bond. The nature of these non-covalent interaction will be described later in the chapter where we will be studying about the chemistry of carbon. The large number of weak interactions between macromolecules in supramolecular complexes stabilize these assemblies, producing their unique structures. Here in this figure, you can see the structure hierarchy in the molecular organization of the cells. In the plant cell, the nucleus is an organelle containing several types of supramolecular complexes, including the chromosomes. The chromosomes consist of the macromolecules of DNA and many different proteins. Each type of macromolecule is made up of simple subunits, which are the DNA of nucleotides or deoxyribonucleotides, for example. So here is the summary of what we have learned today. First of all, we have learned about the biochemical evolution of life, how the life actually began and how the universe was actually formed. And then we learned about different basics of different biomolecules or the foundations of biochemistry. We have learned that all the cells are bound by the plasma membrane. They have a cytosol containing metabolites, coenzymes, organic and inorganic molecules, and they have a set of genes which are contained in the nucleotide in the case of prokaryotes and nucleus in case of eukaryotes. We have learned about different dimensions of lives, and we have seen that phototropes, they use sunlight to work, and chemotropes oxidize fuel. passing electron to good electron acceptors. We will learn about the electron acceptors and donators in detail. We have learned about the inorganic compounds, organic compounds, and the molecule oxygen. We have also learned that bacterial cells contain cytosol, a nuclide, and plasmid. We have actually seen the biochemistry of these molecules. Eukaryotic cells, they have a nucleus and they have a multi-compartmented segregation for certain processes and specific organelles, which can be separated and studied in isolation. We have looked at the structure of the protein assembly in case of in the form of a figure, and we know that cytoskeletal proteins, they assemble into long filaments that give rise to the shape and rigidity and serve as a rail along which cellular organelles move through the cell. We have looked at the supramolecular complexes, which are held together by non-covalent interaction and form a hierarchy of the structures, some visible with the light microscope. When individual molecules are removed from these complexes to be studied in vitro, interactions important in the living cells may also be lost.